Today, we're diving into the amazing world of meiosis. It's all about how cells divide and make more cells. You might be thinking, wait, isn't that mitosis? You're close. Mitosis is how most of our body cells make identical copies. But there's a special kind of cell division called meiosis. It's just for making gametes, sperm and egg cells, with half the usual number of chromosomes. Therefore, they can join to form a new organism. Why is meiosis so important? If meiosis doesn't happen correctly, a gamete might end up with an extra chromosome. This is called non-disjunction, when chromosomes fail to separate properly. If a sperm or egg cell with an extra chromosome 21 fuses with a normal cell, the baby will have 3 copies of chromosome 21, a condition known as trisomy 21, or Down syndrome. Down syndrome was first described as a clinical condition in 1866 by British physician John Langdon Down. But it wasn't until 1959 that French geneticist Jérôme Lejeune discovered the cause, an extra copy of chromosome 21. This discovery marked a turning point in our understanding of genetic disorders. Meiosis has 2 rounds of division, meiosis 1 and meiosis 2. In meiosis 1, we go from 1 diploid cell to 2 haploid cells. As prophase Y begins, thread-like chromatin condenses tightly to form distinct, visible chromosomes. Each chromosome has 2 sister chromatids joined at the centromere. These chromosomes are now ready to pair up with their homologous partner to form tetrads or groups of 4 chromatids. This is also when crossing over, genetic exchange, occurs between non-sister chromatids, which increase genetic diversity. Then, the nuclear envelope breaks down and spindle fibers form from the centrosomes. These fibers attach to the chromosomes, preparing them for alignment and separation. Metaphase 1. Homologous pairs line up along the middle of the cell. Anaphase 1. Homologous pairs are pulled apart to opposite poles. Sister chromatids stay attached. Now we're in telophase 1, the final stage of the first meiotic division. The homologous chromosomes have arrived at opposite poles of the cell, with each side now holding half the original number of chromosomes. But keep in mind, the sister chromatids are still joined at their centromeres. The cell's membrane begins to pinch inward, spindle fibers break down, and new nuclear envelopes briefly form around each set of chromosomes. Cytokinesis is when the cell physically splits into 2. The cytoplasm divides, creating 2 haploid daughter cells, each with half the number of chromosomes as the original. Now let's head to meiosis 2. In prophase 2, nuclear envelopes break down, centrosomes move apart, and spindle fibers form again. Metaphase 2, the chromosomes line up along the center of each cell. Unlike metaphase 1, there are no homologous pairs, just individual chromosomes, with sister chromatids still joined at the centromere. Anaphase 2, sister chromatids are pulled apart as spindle fibers shorten, moving them to opposite poles. Each chromatid is considered an individual chromosome. Telophase 2, chromosomes reach opposite ends of each haploid cell. New nuclear envelopes form and spindle fibers disappear. Cytokinesis splits the cells, producing 4 unique haploid cells. Finally, the chromosomes begin to uncoil, returning to a less condensed thread-like form. Let's recap the highlights of meiosis. Meiosis produces gametes with half the number of chromosomes. It begins with 1 diploid cell, with 46 chromosomes in humans. It ends with 4 genetically different haploid cells, each with 23 chromosomes in humans. It has 2 rounds of divisions, meiosis 1 and meiosis 2. Meiosis is how our bodies create gametes, that's sperm in males and eggs in females. Each gamete is haploid, meaning it carries just 23 chromosomes, or half the usual number. During fertilization, a sperm and an egg fuse to form a diploid zygote with a full set of 46 chromosomes. That tiny zygote is the very first cell of a brand new human life. So, we know meiosis makes sperm and egg cells with half the number of chromosomes. But why is this so important? Well, it's all about variety. Because of meiosis, you are unique. You're not an exact copy of your parents or siblings. That's because you inherit 50% of your DNA from your mother and 50% from your father, but it's not a straight split. As mentioned, during the formation of gametes, a process called crossing over takes place. This mixes up segments of DNA between your parents' chromosomes, creating a completely unique combination of genes. So instead of getting a simple 50/50th slice from each parent, your genetic code is a brand new blend. That's why you might look more like a grandparent, a sibling, or even have features that are totally your own. Sometimes meiosis doesn't go perfectly. When errors occur, they can lead to chromosomal abnormalities, conditions caused by an incorrect number or structure of chromosomes. Like in the Down syndrome, which happens when a person has an extra copy of chromosome 21. Scientists are now exploring ways to understand and potentially address genetic conditions using powerful gene editing tools like CRISPR. CRISPR allows researchers to target and edit specific DNA sequences with precision. While it can't yet fix large-scale chromosomal changes like an entire extra chromosome in Down syndrome, it can help correct single-gene mutations or regulate how certain genes are expressed. A recent study published in PNAS Nexus February 2025, researchers successfully removed extra copies of chromosome 21 in Down syndrome cell lines. By targeting specific alleles, researchers achieved trisomic rescue in both stem cells and fibroblasts, restoring normal gene expression and cellular function. This breakthrough offers a potential pathway for future therapies addressing chromosomal abnormalities.